Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled Continuous Glucose Monitoring and Glycemic Variability in Pregnancy. This webinar has been sponsored by Harvard Bioscience and DSI, so a big thank you to them for helping to make this event possible. Joining us today, we're very fortunate to have Caroline Wuyts, a PhD candidate in the Department of Cellular and Molecular Medicine at KU Leuven. Her presentation will highlight her research on pregnancy and monitoring glucose availability throughout the reproductive timeline of a mouse model. And I'm very excited to welcome our presenter, Caroline. Thanks so much for being here with us today, and the floor is yours whenever you're ready.
Okay, thank you, Sarah. And welcome everyone to this webinar, which is about continuous glucose monitoring. This is a new technique that we've been implementing during pregnancy in mice, which may have important implications to study gestational diabetes. So gestational diabetes is a form of hypoglycemia that occurs for the very first time during pregnancy. And in Belgium, it occurs in one out of 8 pregnancies. The sad thing about it is that it has negative pregnancy outcomes for the mother, which includes an increased risk on type 2 diabetes later in life, but also for the baby, which include that they grow too big, that their shoulder can get stuck upon birth, which means they would opt more often for C-sections, which comes with other complications. And moreover, they also have an increased risk on metabolic disorders later in life. Unfortunately, the molecular mechanisms behind gestational diabetes are not known up until now. And this leads to the fact that there are no specific drugs available to treat this disease. Unfortunately, there are also no good animal models available to study the molecular mechanisms behind gestational diabetes, and the standard techniques that exist to measure glycemia are very limited in resolution. So we decided to use continuous glucose monitoring instead, and I will talk to you about this new technique for a moment first. So generally, what has been a general standard technique that is being used to determine the glucose tolerance is a glucose tolerance test. And in this test, you first fast the mice for 6 hours, then you administer glucose, and then during 5 time points in a time span of 2 hours, you take a blood sample, and in this blood sample, you measure the blood glucose using a standard glucometer, as shown in the top left side of this slide, which is also used by diabetic patients. However, we decided to opt for continuous glucose monitoring, which means that we can have a value for every 10 seconds. So how does this work? You can implant a glucose transmitter in the mouse. This will measure the blood glucose, sends the signal to the receiver. The receiver will go through the matrix and to the computer. And then after calibration, you get glucose values for every 10 seconds on your computer screen. So a telemetric implant looks like this. So in the top you have a glucose sensor, which is implanted inside the blood and which senses the blood glucose, and this sends a signal to the transmitter body, which sends the signal to the receiver. So a glucose sensor contains an enzyme, glucose oxidase, which catalyzes the reaction of glucose and oxygen and will generate a current on the electrode, which is then sent to the transmitter body. So the advantages of continuous glucose monitoring are that you don't have to handle the mice as much, you don't have to take that many blood draws, and there is a severely reduced human presence. And all of this leads to reduced stress levels in the mice. On top of that, you have a tremendously increased amount of detailed data. So you don't only have daytime data that you can generally collect during the working hours, but you also can collect a lot of nighttime data. And on top of that, you can also collect spontaneous glucose excursions that occur when the mouse is just relaxed, non-stressed in its cage. There's also very little influence of the researcher, because whenever the researcher induces stress, the blood glucose levels will increase, and long-term studies are possible. So I will now talk to you about the optimization that we have performed of the transmitter implantation. So generally, the transmitter body is implanted intraperitoneally. However, because we decided to make the mice pregnant, we opt for inserting the transmitter body subcutaneously. Unfortunately, this came with problems that the skin at the subcutaneous pouch ruptured. And this was happening when we implanted the transmitter body after using blunt dissection using scissors. So this results in the loss of the mouse and the transmitter, and we try to prevent this by optimizing the surgical protocol. So first we make an incision at the abdomen. Through here we insert the transmitter body subcutaneously. Afterwards we tunnel the lead to an incision at the ventral neck region, like this. And then the glucose sensor is implanted via the left carotid artery into the free-flowing blood of the aortic arch. So here, the bifurcation that you can see in the middle picture is occluded, and this is used as a reference to insert the glucose sensor in place. So as you can see on the right picture, you can measure, we beforehand measured the insertion depth, and then you can implant the glucose sensor as according to the protocol that is described. So then when the glucose sensor is in place, we also added an extra suture to hold the transmitter body in place. And we use flamazine cream to keep the skin moist and to prevent skin rupture. Another problem that occurred to us is that the stitches open due to the tension on the skin, because the lead is relatively stiff, and the stitches in the ventral neck region tend to open a bit. And therefore, we solve this using Donati stitches, which can bear a bit more of tension. So the optimization of this implantation protocol prevents the loss of the mouse and the transmitters. So then I will tell you a bit more about the protocol that we used to monitor the pregnancy in mice. So in this picture, you can see in the left side a picture of a non-pregnant mice, where on the right side of the animal you can see the transmitter body, and on the right you can see a pregnant mouse, where again on the right side of the mouse you can observe the transmitter body. In general, the transmitter is not bothering the animal. They move after the surgery. They move very fluently and naturally in their cage. So what did we do? We first implanted the transmitter, and then we let the mice recover for at least 1 week, where we monitored their body weight and how they are feeling. And if necessary, we apply extra flamazine, or we give a painkiller. Then there is a multi-point calibration, and we mate the mice. During the full pregnancy, we monitor with continuous glucose monitoring, but we also placed a camera next to the cage, to which I will come later. And then up until 1 week after pregnancy, we keep on monitoring. At the end, we explant the transmitters and we check whether the transmitters were implanted in the correct position. So to test the glucose tolerance, we performed 4 glucose tolerance tests, one before pregnancy, two during pregnancy, and one after delivery. Because we were interested whether the transmitter has an influence on the pregnancy or on the pups, we decided to include a sham group. And in the sham group, they also underwent a surgery where the left carotid artery was occluded and a subcutaneous pouch was made. We compared this to the transmitter mice, and we saw that there was no difference in body weight increase of the mothers. There was also no difference in the litter size and in the birth weight of the pups. And only in the first experiment we saw that there was a decrease in survival of the pups compared to the sham group. However, this is probably due to the fact that Black 6J mice are relatively bad mothers, and their first litter often goes wrong. Further experiments, we didn't observe any problems like that anymore. So this means that telemetric implants are safe to be used in pregnant mice. So then we will move over to the results section, where I will explain some more about the detailed description of the glycemia in pregnant mice, and I will first talk about the glucose tolerance. So, as I said before, we did glucose tolerance tests before pregnancy at embryonic developmental day 8, 16, and 2 days postpartum. So, in a glucose tolerance test, as mentioned before, you fast the mice for 6 hours, we administer glucose, and then we can follow up the blood glucose for 2 hours at least, but of course we could go longer because we have the continuous glucose monitoring. Because we wanted to know whether the continuous glucose monitoring gives us accurate values, we also decided to use the glucometer and measure during every glucose tolerance test the same time points as we would do for a normal glucose tolerance test. And throughout the full pregnancy, before and after, we obtained really good correlations between the glucometer values and the continuous glucose monitoring values. If we then analyze these glucose tolerance tests, we first looked at the area under the curve, and we observed that there is an increase of area under the curve during pregnancy compared to before and after pregnancy. This means that the glucose tolerance is lower during pregnancy in healthy mice. And if we compare this with some data that we have available in pregnant healthy women, we also observe that the area under the curve increases during pregnancy compared to before pregnancy. So in the graphs on the right, you can observe the orange line, which is the peak blood glucose level of the women before pregnancy. And you see that in early and late pregnancy, these values are all higher. But we can also look at the fasting glucose values. And again, we see that the fasting glucose values are increased during pregnancy compared to before and after pregnancy. However, we have found some similar data in women, and here we actually see that the fasting glucose decreases during pregnancy. Of course, it's important to remark that fasting for 6 hours in mice is very different from fasting overnight in women. But of course, so here you can see the oral glucose tolerance test indicated by the red arrows, but we can measure the blood glucose for every 10 seconds during the full pregnancy and even before and after. Because this generates a lot of data, we decided to automate the data analysis. And this is something that I will talk to you about now. So we wrote a Python script to automate the data analysis. And there we looked at 3 different categories. So the first one is parameters that are generally measured using continuous glucose monitoring, or the transmitters that we have available. Besides that, we looked at glycemic variability. And lastly, we looked at the glucose excursions that occur spontaneously. So first, we talk about the general parameters. So the setup can not only measure glucose, but also activity and temperature. It measures the activity by because the receiver contains several points where the signal can be picked up from the transmitter, and the more the mouse moves, the more points there are more points where the signal are being picked up, and you can know the activity of the mouse. So when we analyze this, we can actually observe that the mice are more active during the night compared to during the day, and this is normal because mice are nocturnal animals. And besides that, we also see that the activity during the night decreases the more pregnant the mice are, or the further in pregnancy they are. So besides that, we can also measure the subcutaneous temperature, and this is being measured inside the transmitter body itself, and that's why we would measure subcutaneous body temperature. And here we observe that at the end of pregnancy, so right after delivery, the temperature increases. But then, of course, we can also measure the mean glucose values during the full pregnancy. And if we take the average per 24 hours, we actually observe an increase and then a decrease, where the point where this changes from increasing to decreasing is embryonic developmental day 10. And this is also around the time that the pancreatic beta cells start to adapt to pregnancy and start proliferating. If we measure the baseline of our blood glucose values, you actually observe a similar trend as in the mean glycemia. But we can also take the 5th and the 95th percentile, where the 5th percentile gives a view of the lower blood glucose values, and the 95th percentile gives an overview of the higher blood glucose values. And in this graph, we can see that the 95th percentile actually is less variable than the 5th percentile, which means that the shift of mean glycemia is more because of a shift in the lower glucose values, represented by the percentile, by the 5th percentile, compared to the higher values. So it's more baseline values that will start changing than the peak values. And if we compare these to women, however, there's very little data available, we can also observe that there is an increase from early gestation to mid-gestation, and then that there is a decrease again in average glucose values in healthy pregnant women. So then I would like to talk about the glycemic variability. So if we zoom in, in 1 day and 1 night, we actually observed that, and also in the overall view, you can see that the glycemia is very variable. And we calculated some parameters to quantify this. So you can measure the intraday variability, and we measured long-term intraday variability by taking the standard deviation and the interquartile range. However, you can also measure the short-time intraday variability, and an example of this is CONGA-1, where CONGA-1 is gives a measure of the variability over 1 hour, and the standard deviation and the interquartile range give an idea of the variability per 12 hours or per day or per night. But besides that, we can also measure the intraday variability, where we measure the mean of daily differences. And in general, we see that all types of variability are relatively stable during pregnancy. However, they increase towards the end of the pregnancy, so towards the delivery. And this increasing part seems to start earlier in the daytime values compared to the nighttime values. So if we then again compare this to what there is available in literature in women, we also observe that the glucose variability remains relatively stable during the pregnancy. However, in healthy pregnant women, we don't have any data about what happens during delivery or more towards delivery. So this data is relatively similar. And then lastly, we had a look at the glucose excursions. So again, if you look at this insert, you can observe glucose excursions. And we wanted to know, where do these glucose excursions come from? So for obtaining information about this, we placed the camera next to the mouse cage, and we monitored the mice during the day, but also during the night. And we did this using infrared light. And we did this early in gestation, mid gestation, and late in gestation, every time 1 day, so 24 hours. And if we observed the eating episodes, and we compared this with the glucose excursions, we saw a relatively good correlation. So here you can see the red eating episodes correlate nicely with the glucose excursions. But now we know where these glucose excursions come from. However, we still needed to find a way to analyze them, where the first step is to find these glucose excursions. And for doing this, we first determined the baseline. And this is a rolling baseline, which means that we take, for every data point, we take the lowest glucose value in a time range of 30 minutes around this glucose value. Then we subtracted the baseline, and then we normalized the data for every 12 hours, so for day and for night. After we did that, we placed a cutoff, which was 20% of that highest value for those 12 hours. And now you have starting points and ending points. But as you can see, intuitively, this is not the start and the end of a glucose excursion. And therefore, we started looking to expand our cutoff points towards the beginning and the end of the glucose excursion, where we went looking 20 minutes before our cutoff starting point and 20 minutes after our cutoff ending point. And our conditions were that the slope needs to approximate 0 and that the glucose value needs to be very low. But then you can observe here in the left side that there is moments where you could doubt whether this is 1 or 2 glucose excursions. And therefore, we also decided to join glucose excursions on 2 conditions again, that the end point of the first glucose excursion and the starting point of the second one is closer than 14 seconds, and that all the values between those 2 points are over the cutoff. And then you get this as a result, where every green line is the start of glucose excursion and every red line is the end of glucose excursion. And then we can analyze this by first simply counting the glucose excursions. And here you can observe that there are more glucose excursions during the night compared to during the day. And that actually this difference decreases towards the end of pregnancy and is lost after pregnancy, which was also when the mice were more similarly active. Then you can measure the area under the curve. And in this case, we believe that the area under the curve, so it's a similar approach as what we did in glucose tolerance test. And we could use these values as a proxy for a glucose tolerance test, but when the mice are calmly non-stressed in their cage. We can also measure the time to peak and the maximum slopes. So these give all information about how fast glucose increases and decreases. And we saw that what we call the glucose uptake is lower during pregnancy compared to before and after, and also the glucose clearance, so the decreasing part of the glucose excursion, was also lower. So blood glucose values increase and decrease slower during pregnancy compared to before and after. And then lastly, you can also measure the amplitude of these glucose excursions. And if you then take the average, you calculate the MAGE of the mean amplitude of glucose excursions, which is also a value for the variability. So, as I mentioned, these spontaneous glucose excursions can actually give similar information as a glucose tolerance test. However, the mice are just calmly in their cage, behaving naturally and are non-stressed. So most of the glucose excursions correlated nicely with the eating episodes. However, there are still mostly tinier glucose excursions that did not correlate with the eating episodes. And for those 3 days that we analyzed this, so at embryonic developmental day 2, 10, and 17, we divided the glucose excursions into non-prandial glucose excursions and postprandial glucose excursions. And if we then look at the mean amplitude of the glucose excursions, we observe that there's an increased MAGE towards the end of pregnancy for the postprandial glucose excursions. And this is something that was also observed in women. So after a meal, the glucose values increased more compared to non-pregnant control groups. So I've been showing you the comparison between the glycemic adaptations in mice and in human pregnancy throughout this full presentation. And here is a small overview of them. So in general, we saw that most of the glycemic adaptations are similar between mouse and women, which means that mice may be relevant models to study glycemic adaptations to pregnancy. Then I would like to end this presentation by talking about the end of the pregnancy. So that is the delivery of the pups. Because using the video monitoring and combining this with the continuous glucose monitoring, we could monitor for the very first time what happens during delivery. And so there are 5 example traces on the right side of the screen, where you in general can observe that the glucose values decrease during delivery. Okay, so now I would like to go over some conclusions from this presentation. So first of all, we have optimized the implantation protocol, and this prevents the loss of mice, but also of the implants. Besides that, we have shown that telemetric implants are safe to be used in pregnant mice, and we used continuous glucose monitoring to describe the glycemic adaptations in a lot of detail. Together with the camera surveillance, we could monitor, for example, the postprandial glycemic variability, but also the delivery. And we saw that these glycemic adaptations in mice are very similar as in pregnant women. And all of this together means that continuous glucose monitoring could be used to study and characterize new mouse models in the research that could help in the research for gestational diabetes. So to end this presentation, I would like to thank the full laboratory of ion channel research, and more specifically my promoter, Professor Rudy Vennekes, and my co-promoter, Dr. Koenraad Philippaert, for all the help and the support during this project. And besides that, I would also really like to thank DSI for the cooperation that made this study possible.
Fantastic. Thank you so much, Caroline. That was a really amazing presentation, and I'm really excited to get into the Q&A. We have lots of questions here. The first question is, what diet are these mice on?
Well, before the study, they are just on regular maintenance chow, so it's just a rat and mice chow. And then from the moment they are implanted, we change the diet to a breeding diet, which has some extra nutrients so that the mice can carry on through their full pregnancy properly.
Okay, fantastic. Thank you. The next question here is, did you notice any glucose changes or spikes when the animals were being handled or when people were entering the room that they were housed in?
Yes, so first of all, during the glucose tolerance tests, we also measured manually using a glucometer, because we wanted to know whether there was a good correlation between the glucometer and the continuous glucose monitoring. And actually, in those glucose traces, we see that at time point 1 hour, you can actually observe that there is a small increase, which we do not have if we don't measure the glucose manually. So this small increase there is probably due to mouse handling, so measuring the blood glucose. And then besides that, we also, I mentioned that we did the video monitoring, and I did not only look at when the mice were eating and when they stopped, but I also took into account when they were when the cage was opened, or when someone passed by or stuff like that. And so I also noted this down, and here we also saw indeed that there was an increase in blood glucose. But of course, this increase is a lot smaller compared to when they eat or when you inject glucose, of course.
Okay, that makes sense. All right. The next question here is, for the glucose tolerance tests, what food or substance did you administer to carry out that test?
Yeah. So we injected a 30% glucose solution, and we did it orally, so via gavage. And the dose was 2.5 grams per kilogram body weight of the mouse.
Okay, great. A couple people had that question, so thank you for that. The next question here is, the area under the curve after an oral glucose tolerance test increases with gestation. Glycemia, however, increases and then decreases. Are you able to explain that?
Yeah, that's indeed true. Well, then we can think of the important difference between a glucose tolerance test and when the mice are just calmly in their cage. So during a glucose tolerance test, you first of all you inject a high glucose dose, which is higher than what the mouse would eat normally, and you also handle the mouse, so you administer the glucose, which is already completely different from the continuous glucose monitoring, so the average glycemia that we took while the mouse was just calmly in their cage. So maybe that's where the difference comes from. And then it might be that so when the mouse is given a lot of glucose, there is a worse insulin or a worse insulin sensitivity compared to the lower glucose values that occur normally in the normal behavior of the mouse. So maybe that's where the difference comes from.
That makes sense. All right. The next question here is, have you done this in mice that have undergone many pregnancies? And if so, how does one pregnancy compare to many? And also a very interesting talk.
Yes, so initially we started doing this in mice that only underwent a first pregnancy. That was their first pregnancy, so they were virgins before we implanted the transmitter. However, later on we also now we're using mice that already went through a first pregnancy. And sorry, what was the second part of the question again?
The second part of that question was, how does the data that you collected differ between a single pregnancy and then a mouse that has undergone many pregnancies?
Yeah. Yeah, so we've never measured continuous glucose monitoring of 2 consecutive pregnancies in one mouse. So unfortunately, I can actually not answer that question. Yeah, it's
That's okay.
it's possible that there's indeed a difference. Mainly, yeah, there's a lot of differences between the first pregnancy and further pregnancy, mainly for black 6 mice that are not too good mothers. However, I cannot answer that in the same, because we haven't done it, the continuous glucose monitoring. It would take too long. It would not be possible to measure more than one pregnancy consecutively.
That makes sense. Okay, related to mothering behavior, Did you notice the implantation of transmitters changing the outcome of the pregnancy and how good of a mother the mice were after they delivered?
Mm-hmm. Yeah, so we had a look at indeed. So we compared the transmitter mice with the sham mice, and the pregnancy on its own, and the pups and the litter. So we saw that there was a same amount of pups born, and they had the same body weight. So during pregnancy, they probably have very, very little influence of the transmitter. And then after pregnancy, so after delivery, there was one group that really had a worse survival of the pups. However, this was really only one time that we saw this. It might be due to the fact that the lead is subcutaneously, so maybe there is less. So there's less nipples available for breastfeeding. However, it's only one time that we saw this, and all the experiments that we have performed afterwards, we didn't see any more mortality in the pups from the mothers with the transmitter.
Okay.
Yeah, and also as I said, Black 6 mice are really bad mothers for sure for the very first time in the pregnancy, when they were pregnant.
Okay. All right. So the next question here is, how long is the delay between consuming food and then the associated glucose spike? And is it different between pregnant and non-pregnant mice?
Mm-hmm. Okay, so I did not exactly measure how much the delay was between consuming the food and the spike. However, what we did measure was the time to the peak for the glucose excursions,
Okay.
which is around 16 minutes, which is actually very similar to what you see in a glucose tolerance test. So in a glucose tolerance test, you also administer glucose, and then usually around 15 minutes or a bit earlier even, you can see the peak. What we did see, what we also measured, was the glucose increase, so the glucose uptake, and the glucose decrease, so the glucose clearance from the blood. And we observed that this was slower during pregnancy. So in that sense, you could say that the glucose excursions, so the glucose dynamics, were slower. And then before and after pregnancy, the glucose excursions were more sharp.
Okay, so it's just more of a delay in the clearance of glucose and everything during pregnancy. Okay,
yeah.
That makes sense. Do you know if that is similar in human pregnancies as well?
I do not know that, because unfortunately there's very little data available in pregnant women, or in healthy pregnant women, moreover. But as I also mentioned during the presentation, you can observe that the amplitude goes higher during pregnancy in pregnant women. So right after eating, the amplitude of the glucose excursion is higher, which we also saw in our mice.
Okay, interesting. Cool. Thanks. That was just a personal curiosity. All right, we have another question here. Did you have any issues with the implant no longer collecting data because of a long latency? So basically, did you get the full course reading from implant to postnatal for all of the mice that were implanted?
No, indeed, some mice, for some reason, really take a lot of time to get pregnant. For most mice, they get pregnant very fast. So within the first week. However, some of them really they took 2 weeks to get pregnant, and then indeed you miss already 2 weeks while the transmitter is implanted. And sometimes the transmitter still makes it, because the transmitter can really measure for a very long time. But not for all mice. This didn't work out for all mice. So indeed, for some mice, we don't have the full 29 days of data.
Okay. And the recovery period from implantation to when you put them in the breeding cages, how long is that?
We take around 1 week. So we implant the mice, and then we wait at least a week where we really follow them up daily. We measure the body weight, so on and so on. Usually after 6 days, they are fine already, but we still wait an extra day to be sure. And then if sometimes a mouse needs a little bit more time, we also give it to them, because it's important that they're healthy before you start the study, of course. But normally it's around 1 week. Maybe one day a mouse takes 8 days instead of 7. But
okay, makes sense. Thanks. We've got a couple more questions here. So this next question is, what is the time resolution that you're using to record for the extent of the pregnancy? So how often are you sampling?
We have a blood glucose sample every 10 seconds.
Okay, yeah. So it's a pretty tight timeline.
Yeah, so we have blood glucose for every 10 seconds, and that during 29 days long. Yeah, so it's quite a lot of data.
Yeah, it's pretty comprehensive. How long does it take you to analyze all of that data?
As I mentioned, we have the Python script that we wrote. And when we have that, it just takes, you just insert the data there, and then you let it run. It usually takes a good weekend, maybe a bit longer, so 2-3 days for all the mice. So that doesn't take that long. So we're very happy that now we have the Python script, and the analysis can go very fluently.
Yeah, that's amazing. And yeah. So the only part that takes you a really long time is really the video monitoring. How long does that take to analyze?
Yeah, that takes a lot longer. Unfortunately, we haven't come up with a solution for analyzing that. But that takes, I would say, you can, of course, you can make it go faster a bit while the mouse is sleeping, for example. So you can do around 36 hours per workday.
Amazing.
Sometimes it goes a bit faster, sometimes it goes a bit slower.
Yeah, that's a lot of data to watch and analyze. Are you doing that all by yourself?
Yeah, so far, yes. But so that's why we also decided to do only 3 days during the full pregnancy.
Okay, that makes sense. Awesome. All right. We do have another question here. This question is a little more philosophical. This person has asked, as continuous glucose monitoring use is becoming increasingly popular in human pregnancy, what new and significant data do you think we can achieve from utilizing this technique in animals that we can't in human women?
Yes, it's becoming more easily available. However, they're not going to use continuous glucose monitoring for gestational diabetes yet. So indeed, for type 1 diabetic patients, it's used more often already. But unfortunately, well, yeah, for continuous for gestational diabetes, it's not used that often. Also because it's often only well, it's only tested at the end of pregnancy. So then the time span that they would have this device for is very short. So still there is a lot of information lacking there. Well, and then of course, as a second point, we cannot perform studies on pregnant women. There are very strict ethical questions there. So why this continuous glucose monitoring in pregnant mice is so useful and important is because we can look, first of all, we can test several drugs. Well, actually, first of all, we can have a look at genetically manipulated mice, so knockout mice or knock-in mice or whatever you want, and whatever it can be existing. And then second of all, if we would find a good animal model for gestational diabetes, we would be able to start testing drugs, and in that way get more information about what may help in pregnant women.
Mm-hmm. Awesome. Yeah, I guess it does make sense having someone, a normal, healthy human pregnant woman wouldn't necessarily go to the doctor unless she was experiencing symptoms. So you
Yeah,
don't have
no,
that like healthy data, which makes sense.
yeah. So in Belgium at least, but I think in most countries, every pregnant woman gets tested, receives an oral glucose tolerance test at the beginning of the third trimester.
Yeah, I think that's standard here in Canada as well. So
Yeah.
that makes sense.
Yeah,
All
so it's always
right.
at the end of pregnancy.
That makes total sense. All right. Just looking at the time here, I think I'm going to make this my last question. This question is regarding your Python script. This person wants to know if you have made it open source for people to use.
So far, it's not open source, but we would be happy to share if people get in contact with us, so that we know what for and why, and maybe make some adaptations together for the use that the person wants to use it.
Great.
So we're
Yep.
always
So,
sorry,
no,
we're
that's
happy
okay.
to collaborate and help people out using our Python script.
So if you're interested in accessing that open source Python script, please just let us know in the Q&A and we'll make sure to pass that information on to Caroline and she can get in touch with you via email.
Yeah.
And I totally lied. I have one last question for you. This is a personal curiosity question. Where do you see the future of this research going?
So as I mentioned already, I really think we should invest in finding good animal models for gestational diabetes. So if so in our case in the lab we have a candidate which might be a good animal model for gestational diabetes, and once we have tested this, we might be able to test drugs and so on.
Fantastic. Okay, well, I just really want to thank you for all of your insights today, Caroline. It really was such a pleasure to have you with us.
Thank you very much.